on, like, through the view of biology, chemistry, and practical medicine. Antibody-proteases. New targeted therapies for autoimmune and chronic inflammatory diseases would require greater understanding of a patient and obligatory a pre-illness person at risk to get the therapy personalized for either of those subsets. In this sense, the identification, impact, and implementation of diagnostic, predictive, and prognostic biomarkers becomes the holy grail of platforms, algorithms, and protocols which are the crucial for personalized and precision medicine. So it's necessary to identify biomarkers of the newer generation to create a fundamentally new strategy based upon subclinical, predictive recognition of the biomarkers long before the disease clinically manifests itself. Among the best validated predictive biomarkers are autoimmunity-related ones to predict and to prognosticate risks of the chronification complications and thus disabling. Just an example of those biomarkers gifted by my very close friend, Yehuda Schonfeld. It's a dean of medical school from Tel Aviv, Israel University. Since chronic autoimmune diseases are preceded by a long subclinical symptom-free phase, look, normal immunity, benign autoimmunity, stage of subclinical autoaggression, pathogenic autoimmunity, stage of full-term autoaggression, and finally, clinical illness. Along with canonical antibodies, some of the families proven to occur are antibodies armed with catalytic activities, so-called CAT antibodies or enzymes. Antibody, enzymes. And thus to belong to antibodies with the feature of functionality. Functionality is a catalytic properties. Look, catalytic antibodies are multivalent immunoglobulins presumably of IgG isotype, endowed with the capacity to hydrolyze the specific antigen substrate. The property mentioned, I mean the catalytic property, is buried in FAB fragment of IgG molecule and is appearing to sound as a functional property of the AB antibody molecule. In this sense, proteolytic antibodies, so-called antibody-proteases, as an example of enzymes, as a significant portion of the big family of enzymes, represent antibodies endowed with the capacity to provide targeted protein effect. Other types of enzymes, DNA, RNA, and others. That's the hypothesis, how enzymes are being formed. If you could do it, please read yourself. It's a kind of transition state analysis. A situation of phenomenal interest is occurred in multiple sclerosis, which is a chronic autoimmune inflammation in central nervous system, CNS, resulting in a destruction of myelin by different tools and leading to demyelation, axon loss, and development of disability. The crucial step in the pathogenesis of MS is the primary myelin damage mediated by autoantibodies to trigger a release of separate and pathogenically valuable myelin-associated autoepitopes in the bloodstream. Anti-myelin antibodies damage myelin. A spectrum of myelin-associated autoantibodies occurring in MS patients have been confirmed to be very large. It's just an example of the main ones. One of the crucial MBP, myelin basic protein. We have established that anti-MBP autoantibodies from MS patients and mice with MS analog experimental autoimmune encephalitis exhibited MBP-specific proteolytic cleavage at the MBP molecule. The activity of antibody-proteases markedly differs between MS patients and healthy controls. Also differs in MS patients with different causes of MS. High levels and high activity in aggressive forms. Exacerbation of remittent and progression of progression. Lowest activity in less aggressive. Remission and stabilization. And finally, the activity demonstrated significant correlation with scales of demyelination, neurological deficiency, and disability of the patients. It's a special MS score illustrating the latter. Of greater value is this sequence specificity. of antibody-proteases, which was analyzed by screening peptide cleavage products by a combination of reverse phase chromatography, SDS electrophoresis, Western blot analysis, and mass spectrometry. It's just the technology. Antibody-mediated proteolysis of MBP in MS patients results in generating a set of peptides with molecular masses ranged in various but fixed boundaries to suit common principles of molecular architectonics of MBP. The final statistical data revealed 5 sites of proteolysis. 1, 2, 3, 4, 5. Most of those sites are located within 43170 region of MBP, which are immunodominant. Two sites from the set located within this sequence, which comprise 4298 and 42103, subsequences that, whilst being sequence-specific, highly immunogenic and encephalotogenic both as well, proven to be specific and major inducers of very autoaggressive MS analog in mice. Moreover, both short subsequences have been proven to be major MBP targets to be attacked by antibody proteases in MS patients with mostly aggressive courses of MS, including secondary progression phase and remittent courses exacerbation phase. The most immunogenic and immunodominant sites responsible for generating aggressive bursts of demyelination are concentrated in 3 major areas of MBP molecules. Strongest one in the smallest subsequence, weaker one in the shorter one, but another one. A site with the lowest immunogenic properties is formed by rather long subsequences. The final step of the studies concerned the evolution of antibody-associated proteolytic activity at different stage of the disease progression, including subclinical and clinical ones. For those purposes, we have been monitoring MS patients, their direct relatives, and healthy voluntaries for around 2-3 years. As you see, apart from MS patients with the diagnosis confirmed, 72% and 18% of the relatives were initially seropositive for anti-MBP autoantibodies with no proteolytic activity, so-called disarmed antibodies. And MBP specific but low active antibody proteases, respectively. Neither of those seropositive relatives, regardless to type of antibodies, demonstrated neither clinical manifestation nor instrumental ones of clinical MS. The relatives being seropositive initially for antibody proteases were being monitored, demonstrating a stable growth of MBP-targeted antibody-associated proteolytic activity. And you see, in 6 months, in 12 months, the growth, in 2 years, in 3 years. In 3 years, almost like in aggressive forms. Along with the evolutionary changes of the sequence specificity of antibody proteases, we could predict transformation from subclinical into clinical course, changing of a remitting type, moderate one, into the secondary progredient type, severe one, prior to changing of clinical manifestation, subclinical indication. So, sequence-specific antibody proteases have proved to be greatly informative and thus valuable as biomarkers to monitor MS at both subclinical and clinical stages. The translational potential of this knowledge in the rational design of new diagnostic tools and new targeted therapeutics based on principle of artificial biocatalysts and biodesign, and to exploit the role of the key pathways in influencing the disorder. The traditional goal of antibody engineering is to combine various antibody domains to generate customized antibodies that show specialized binding properties, optimal half-lives, and desirable effector functions. Antibodies can be engineered to make proteins of higher affinity or smaller molecular variants that retain or change the functional property of the original antibodies. The last years have seen a major upturn in the fortune of therapeutic antibodies approved for clinical use. It's just a scope of antibody applications in practice. Antibody-based therapeutics have entered the central stage of drug discovery as a result of a major shift in focus of many biopharma companies. So, antibody protease design-driven engineering would offer the ability to enhance or alter their sequence specificity to expand the clinical utility of absolutely new tools. In this sense, antibody proteases can be programmed or reprogrammed to suit the requests and standards of regeneration and remyelination. The latter would suit the needs of the body turnover, or could be designed for the development of principally new catalysts with no natural counterparts. Of tremendous value in this sense are antibody proteases directly affecting the physiological remodeling of tissues with multi-level architectonics, for instance, myelin, while securing the request and standards of metabolism. In this sense, targeted antibody-mediated proteolysis could be also applied to isolate from Ig molecules catalytic domains directed against encephalopatogenic autoepitopes or domains containing segments to exert targeted proteolytic activity. Meanwhile, the next important step in the direction of the innovation-based approach should be their early adoption in clinics for future medical interventions. And having my speech summarized, I would stress that we, our international team, have done something to have the first bricks laid into the foundation of the projects of the next step generation, uniting basic research, translational application, clinical studies, industrial pipeline, and finally, a market of the future. Thank you again for your patience.